The nervous system is made out of many different types of cells, but the two main types of cells that you're going to find in the nervous system are neurons and neuroglia. As we've talked about before, the neurons are really the stars of the nervous system. They're the ones that actually do the communicating, that do the actual job of the nervous system. But like all sorts of stars, they require a lot of help. And for every one neuron, there's going to be many, many neuroglia. In this lecture, we're going to talk about the neurons and the different types and classes and structure of the neurons of the nervous system. So, what you see here, this is your standard neuron. Turn to page 1 of any neurology textbook. You're going to find this picture. Nevertheless, as we're going to see, there are many different variants on this general structure, but this is the general structure of a neuron. It has 3 main parts. The cell body, or soma. This is the area that has the nucleus, the cytoplasm, most of the mitochondria, pretty much all of the organelles in the neuron. This is where most of the stuff gets made, whether you're talking about neurotransmitters or proteins or, energy or what. Most of that gets made in the soma. You then have the dendrites, which you see here sticking out from the soma and branching many, many times. Now, there's many different ways that dendrites can look, but they all basically have the same functionality. This is the main area where the cell receives information. It could be receiving information from other neurons. This type of neuron that you see pictured here would almost certainly be being stimulated by another neuron, because this is a motor neuron. We'll talk how you can tell about that in a second. But the dendrites could be covered in connective encapsulations if they were part of the sensory system. It could be connected to other neurons in the brain if it was an interneuron, anything like that. Generally speaking, the more dendrites that a neuron has, the more information it can receive from the more different places. One neuron might form a single connection with the dendrite of another neuron, or it might form hundreds of connections with hundreds of dendrites of another neuron, or it might form hundreds of connections with the dendrites of hundreds of other neurons, depending upon the sort of network that's being set up. The placement of the dendrites actually does matter. And we'll talk about why and how this is in a moment, but suffice it to say that dendrites placed back here, far away from the axon, have less influence than dendrites that are placed very close up to it. The third general area is the axon, which is a long, narrow projection which can lead from the soma to pretty much anywhere. The longest cell in your body is a neuron, and its axon goes from basically the back of your head all the way down to the tip of your toe. So axons, and that's all one cell. That's not even all an entire cell. That's just the axon. So axons can be hugely long. And think about in big creatures like an elephant or a whale or something like that, their axons still span the entire length of their body. It's kind of crazy how long these things can be. The axons have a few important features here. First off, you have the area where the axon joins into the soma. This is called the trigger zone, and it consists of the axon hillock, which is this area that narrows down where the axon is becoming, and the initial segment, which is a very small area of axon that is uncovered, unmyelinated, before the true myelinated axon begins. This is called the trigger zone because this is where the cell decides. It's going to receive information on its dendrites, and that information is going to be processed, and then the cell has to make a decision. Is the axon going to fire an action potential or not? Action potentials are all or nothing. You either fire one or you don't. And that decision is made in the trigger zone. When we talk about action potentials later on, we'll talk about threshold potentials, similar to how we did in muscle cells. Where that threshold potential matters is in the trigger zone. Suppose that this nerve cell does decide to trigger an action potential. That action potential will then propagate down the axon, and let's take a look at some of the features of the axon. First off, the axoplasm is the cytoplasm of an axon. Axons are very small. They don't actually have a whole lot of cytoplasm. The axolemma is the membrane of an axon. And just like sarcolemma was the membrane of a muscle fiber, axolemma is the membrane of an axon. You see these cells here that look like little fleshy pill-like things that are wrapped around the axon. Those are the myelin sheath. And in the central nervous system, they would be made from oligodendrocytes. This neuron, as I said, is a motor neuron. And one of the ways you can tell is because its myelin sheath is made from Schwann cells. And Schwann cells are what myelinates the peripheral nervous system. While a cell, a neuron, can have many, many dendrites, it has only ever one axon, or less. There are actually a few different neurons, a few neurons that don't have axons. We'll talk about those in a second. But the typical rule is one or none, at least to start with. Axons can and do branch. Usually during the main part of an axon, it's traveling really, really far, it's myelinated, and it's mostly going down a totally straight pathway. But as you can see here, there's a branch point off that's called an axon collateral. And you can have some collaterals branch off early on. Often what these collaterals will do is they may actually reach back and suppress this neuron so that you get like a burst of activity and then it turns itself off to kind of like create contrast, sort of like the way that a strobe light is going to have a lot more impact if it's in a dark room than if you have a strobe light in a really bright room. These nerves want to be noticed, so they want to go from like silence to activity, and then boom, turn themselves off. They also may suppress nearby neurons because, again, they want to be noticed, and so they don't want to be mixed up with a bunch of nearby signals. And that's often what collaterals do. In between the myelination, the myelinated portions, the Schwann cells in this case, you have what are called nodes of Ranvier. That's this bit right here. They're tiny unmyelinated sections in between the little patches of myelination. We'll talk about exactly why these things are super important in a later lecture. But suffice it to say that without these nodes of Ranvier, the signal would be unable to propagate down the nerve cell. These are where the signal is renewed, which allows the signal to move basically as far and as fast as it can down the axon. The myelinated portion, by the way, are called internodes, meaning in between the nodes of Ranvier. At the end of the axon, you reach the what's called terminal arborization or arborization. Arbor means tree or forest. So really what this just means is end branching. Terminal means end, arborization means branching. This is the end branching. And you can see that you might have like a couple of main splits that maybe go off and stimulate totally different neurons. But the closer and closer you get to the end of the axon, you get more and more and more branches. And depending upon the type of neuron that you're talking about, it could be hundreds and hundreds of branches at the end of this, each of which is going to end in a synaptic knob. And this being a motor neuron, all of these synaptic knobs are likely to end on a muscle fiber synapse and stimulate that muscle fiber to contract. If this was sensory or an interneuron, it could be that each of these synaptic knobs is going to junction with a dendrite. In any case, all of these synaptic end bulbs, the end bits at the end of the arborization, are full of neurotransmitters that are going to activate whatever is next in line. Now, like I said, the neuron that I showed you on the last slide is your stereotypical page 1, this is your neuron. But there's actually a lot of different variations on that theme. Here are 4 main variations that I want you guys to know. And there are sub-classifications of each of these, but these are the ones that I want you guys to focus on. First, and this is what I showed you on the last page, is a multipolar neuron. In this case, polar refers to how many projections come off from the soma. Here, we see lots of projections coming off from the soma. You've got a dendrite branch here, more dendrites here, more dendrites here, more dendrites here, and the axon over here. They'll have one axon, and usually lots of dendrites coming off of them. This is the most common type of neuron. Most of the neurons in the central nervous system are multipolar. And most of the neurons in your somatic motor nervous system are also going to be multipolar. These neurons can receive information on any of their dendrites and send it down the axon. Next, we have a bipolar neuron. That means that the soma has 2 projections coming off of it. One will be a stalk that will usually lead to a group of dendrites, and the other will be the axon that leads to the terminal arborization and junction with whatever down the end of the road. These are usually found in the, there's usually sensory neurons, not always, but usually sensory neurons in the special senses. So the senses that are only going to be receiving information from one source or place. So you find these in olfactory neurons, where this stock of receptors would be ending in your nose, and then this stock of synaptic end bulbs would be junctioning somewhere in your brain, in your olfactory, probably in your olfactory section of your temporal lobe. You also find them on in the retina for visual receptors, in the inner ear for auditory and balance receptors, and those sorts of things. They're usually very highly specialized neurons. Down here, we have a unipolar or pseudo unipolar neuron. It's unipolar because you can see here the soma has a single projection coming off of it. That projection connects to the axon, effectively. Over here, we have the dendrites, which are usually going to be receiving information from sensory input. This would be the axon hillock right here, and a signal would propagate down the axon to the axon termini without ever passing the soma, right? It's just going to go boom right to the side of the soma. The soma still makes all of the proteins and stuff like that, but has to export them down this projection and either way. This is what you find in most of the sensory neurons of your peripheral nervous system. All the stuff that's going from your skin and your organs, your nociceptors, your thermoceptors, your touch receptors, all of those are going to have little sensory bits at the end of their dendrites and then send information down the axon, where it will junction usually in the spinal cord. And these somas off to the side, usually going to be found in the ganglia. These are called pseudo-unipolar because they actually start off as bipolar neurons in development, and then the soma sort of moves off to the side. So they don't start as unipolar, hence they're sometimes called pseudo-unipolar. The last is an anaxonic neuron. That means that it doesn't have an axon. And here you can see this neuron. It's got dendrites going off in all directions, but no axon. These are, for the most part, found in the central nervous system. There are a few other places where they can be found, such as the retina and the adrenal gland, but most of them are going to be in the central nervous system, and they sort of like glue signals together. They play an interesting role in signal propagation and signal spread, but not one that we are unfortunately going to be able to get into in depth. So that's the different types of neurons. What you need to know is the features of your standard neuron, soma, dendrites, axon, what an axon collateral is, what a node of Ranvier is, myelination, terminal arborization, as well as the 4 types of neurons: multipolar, bipolar, unipolar, and anaxonic. and generally where each would be found.